Hello everyone, welcome to Agri Addict YouTube channel. I hope you all are doing well. So, this is the third chapter of crop physiology lecture series, that is respiration in plants. So, let's start with the lecture. Before starting the lecture, this is my short intro. I have done my undergraduation from JNKVV Jabalpur. Currently pursuing masters at IRI Hyderabad Hafreda Department Agronomy, and I have secured All India rank 12th in ICR PG 2024 examination. So let's start with the lecture. So the first thing we have to know is what is respiration. Okay. So it is the breaking of carbon-carbon single bond of complex compounds through oxidation within the cell, leading to the release of considerable amount of energy. So in respiration, what is happening? Here complex compounds are broken down into simpler compounds, and in this process there is a large amount of energy which is getting released. Okay, the trapping of this energy for synthesis of ATP. Here this energy will be used to synthesize ATP, that is adenosine triphosphate, that is the energy currency of the cell, and this process is known as cellular respiration. During cellular respiration or oxidation within a cell, all the energy contained in a compound is not released in a single step. This respiration is a multi-step process. Here we'll not get all the energy released in one step only. If that is going to happen, then what will happen? All the energy will be dissipated as heat. Okay, so we won't be the plant won't be utilize that energy in the growth of the plant, for the growth of the plant. Okay, so what happens, this energy is not released in a single step. It is released in a series of slow stepwise reaction controlled by enzymes, and it is trapped as a chemical energy in the form of ATP. ATP, the synthesized, is broken down whenever and wherever energy needs to be utilized. Hence ATP acts as an energy currency of the cell. Just like we use currency for buying anything, so plants also use ATP as energy currency. So whenever the plant needs have to divide cell, it needs energy. So that ATP will be used for that. Okay. So the respiration is an exergonic process, means here that energy will be released during this process. Complex molecule will get converted into simple molecule, and during this process the energy will be released. And it is an amphibolic pathway, means it is a process in which anabolism is also happening with catabolism, means something is getting synthesized also and something is getting broken down also. So it is an amphibolic pathway. So let's move to the next slide. Now comes the types of respiration based on the presence or absence of O2. So aerobic, as the name suggests, it takes place in presence of oxygen, whereas anaerobic or fermentation, it takes place in absence of oxygen. So in aerobic respiration, this accounts for complete oxidation. Anaerobic respiration, there is a complete oxidation, means glucose will be completely broken down into carbon dioxide and H2O, that is water molecule. Whereas in case of anaerobic or fermentation, here it is a partial breakdown of glucose. This glucose may be converted into lactic acid or ethanol plus CO2. Lactic acid when there is lactic acid fermentation, ethanol when alcoholic fermentation is there. Now it is an intermolecular respiration, whereas anaerobic respiration is an intramolecular respiration. Here 36 or 38 molecules of ATP is formed from each glucose molecule. Here only 2 molecules of ATP is formed. Because it is a complete oxidation, here we are getting either 36 or 38. Why there is a difference of 2 ATP, we'll study in the later on lecture. And here 2 molecules are formed because it is a partial breakdown, okay, partial oxidation. Here NADH is oxidized to NAD plus vigorously, and here it is oxidized rather slowly, okay. Here O2 remove hydrogen from the system and act as a final hydrogen acceptor. Oxygen act as a final hydrogen acceptor, whereas in case of anaerobic or respiration or fermentation, O2 is absent. So hydrogen acceptor in the system may be acetaldehyde in case of alcoholic fermentation or pyruvate in case of lactic acid fermentation. This you have to remember. Now comes the reaction. This glucose in presence of O2 converted into CO2 and H2O and 686 kilocalorie energy is released. Okay, whereas in this case, in case of alcoholic fermentation, ethanol is formed. In case of lactic acid fermentation, lactic acid is formed and less than 7% of energy of glucose is released in this case because it is a partial oxidation. So let's move to the next slide. Now comes the steps of cellular mechanism of cellular respiration. So four steps are there. First is glycolysis, link reaction, Krebs cycle, and electron transport chain. So what are the site of these uh, process? Like glycolysis occurs in cytoplasm, okay? Whereas link reaction occurs in mitochondrial matrix. Krebs cycle occurs in mitochondrial matrix, and electron transport chain takes place in inner mitochondrial membrane. Okay? Now next. Let's come to the structure of mitochondria, where these all the four procedure is taking place, all the four processes are taking place. It is the mitochondria where it is taking place. So it is a semi-autonomous molecule, okay, and this is also known as powerhouse of the cell. Powerhouse of the cell. It is the site of respiration, okay. So different, like glycolysis is taking place in cytoplasm, whereas all the other three link reaction, Krebs cycle, and electron transport chain, they are taking in this mitochondria at different places. So this is a double membranal structure. First, we have to remember that it is a double membrane structure, just like chloroplast we studied in photosynthesis. That was also a double membrane structure. So wait a minute. So this is the outer membrane. This is the perimitochondrial space between the outer and inner membrane. This, and here is the inner membrane. This is the matrix. Inside the inner membrane, whatever the substance are there, it is in the matrix. That is the matrix. Now it is circular double-stranded DNA because it is a semi-autonomous molecule. It is having its own a double-stranded DNA. Okay, the structure. This is the cristae. It is the enfolding of inner membrane. Cristae. It is increasing the surface area for reaction, and it is also having 70S ribosome. And on this cristae, F0, F1, F0 particle or oxysomes are present. Okay, so I hope the structure of mitochondria might be clear for you. So let's move to the next slide. Now comes the first process, that is glycolysis. So glycolysis is also known as EMT pathway. Why we are calling it EMT? Because the three scientists who found out this process is Emden, Meyerhoff, and Parnas. So three scientists' name you have to remember. And the site of glycolysis is cytoplasma. This you have to remember. And it is a common pathway between aerobic and anaerobic respiration. Whether the cell is undergoing aerobic respiration or anaerobic respiration, irrespective of that, glycolysis will be there for aerobic also and anaerobic respiration also. Here, no CO2 is released, no O2 is released. Okay, it is an incomplete breakdown of glucose. This you have to remember. And here, one glucose molecule, which is a 6-carbon compound, is broken down into 2 molecules of pyruvic acid, which is a 3-carbon compound. Okay, it is a 10-step pathway, and here the energy used is 2 ATP. The first 1 ATP will be utilized in first step, and then again 1 ATP will be utilized in the third step. Now comes the energy produced. Here, the first time the energy will be produced in step 6, that is 2 NADH2, 2 molecules of NADH2. In step 7, 2 ATP, and in step 8, 2 ATP. And this, the 4 ATP which is being produced in this glycolysis process is formed via substrate level phosphorylation. What is substrate level phosphorylation? So substrate level phosphorylation, when ATP is formed without ATP synthase enzyme. As we studied in photosynthesis, there was this F1 F0 particle. Okay, this was there. In that, the H plus was passing and this was rotating, and from stroma, this ADP was combining with inorganic phosphate and was forming ATP. Okay. So if the ATP is not formed using this ATP synthase, then that kind of phosphorylation is known as substrate level phosphorylation. So the 4 ATP which we are getting here in glycolysis is substrate level phosphorylation, because here the ATP synthase enzyme is not being used. Okay. So let's move to the next slide. So here the net energy we are getting in glycolysis is 2 NADH2 and 2 ATP. Why? Because we are utilizing 2 ATP, okay, and we are producing 4 ATP. So 4 minus 2, only 2 ATP is left. Okay, so here only net 2 ATP are formed and 2 NADH2. So if we will calculate, because NaDH2 is equals to 3 ATP. So it will calculate 2 into 3 would be 6 plus 2, 8 ATP is the net energy which will be formed from 1 glucose in glycolysis. Okay, here I have written 1 NADH2 is equals to 3 ATP. Now the whole reaction, if we summarize the glycolysis in one step reaction. So here glucose 4 ADP plus 2 inorganic phosphate plus 2 NAD plus molecule plus 2 ATP. Enzymes in cytosol, cytosol is nothing but cytoplasm, is forming 2 molecules of pyruvic acid, 4 molecules of ATP, 2 NADH2 and 2 ADP. So this is the whole reaction, the glycolysis. And here in mature RBCs and sieve tube cells, it is the main energy producing pathway. This you have to remember. Now let's study the process in detail. Okay. Okay. So first, wait. First glucose is there. Hmm. Glucose is there, which is a 6 carbon compound. Here one ATP is being used in presence of hexokinase. This glucose is getting converted into glucose 6-phosphate. Okay, one phosphate is added to the 6th carbon of glucose, and this phosphate came from this ATP which is being used. Okay, now this glucose 6-phosphate in presence of isomerase enzyme is getting converted into fructose 6-phosphate, which is also a 6 carbon compound. Now one more ATP is being used in presence of phosphofructokinase, and this new molecule is formed, that is fructose 1,6-bisphosphate, means fructose having 2 phosphate at first carbon and second at 6th carbon. Okay. Now this fructose 1,6-phosphate either can get converted into this glyceraldehyde 3-phosphate in presence of aldolase, or it can convert into DHAP, that is dihydroxyacetone phosphate, which is a 3 carbon compound. Means when fructose 1,6-bisphosphate molecule will get broken down, it will form 2 molecules of either glyceraldehyde or 2 molecules of either DHAP, that is dihydroxyacetone phosphate. Okay. Now these 2 molecules of glyceraldehyde 3-phosphate in presence of dehydrogenase will use 2 NAD plus and form 2 NADH2. Okay, this 2 NADH2, this is the first time energy which is being formed in the glycolysis, because before that we saw that energy was utilized. But here in this case, Here in this case, the 2 NADH2, that is energy, is being formed in at the sixth step. This is the sixth step. Wait. Okay. So this is the first step. This is the second step, third step, fourth step. Okay. This is fifth step and this is sixth step. So at sixth step, we are first time getting this energy in the form of NADH2, that is two molecules of NADH2. Now this glyceraldehyde 3-phosphate is getting converted into 1,3-bis-phosphoglyceric acid, which is a 3-carbon compound. So here two molecules will be formed of 1,3-bis-phosphoglyceric acid. Again, 2 ATP will be used and here 2 ATP will be formed at the seventh step in presence of phosphoglycerate kinase enzyme. These enzymes you have to remember at which step which enzyme is being used, because in JRF examination they can directly ask the enzymes which is being which is playing role or catalyzing the reaction. Okay, now this 1,3-bis phosphoglyceric acid will get converted into 3-phosphoglyceric acid, two molecules of 3-phosphoglyceric acid, and this 3-phosphoglyceric acid in presence of mutase enzyme will form 2-phosphoglyceric acid. Okay, here also the two molecules will be there. Now in presence of enolase, two molecules of H2O will be released and 2-phosphoenol pyruvic acid will be formed, and here also two molecules will be formed. Now again 2 ATP will be formed while converting this 2-phosphophenol pyruvic acid in presence of pyruvic acid kinase enzyme. This will form 2-pyruvic acid, that is the last end product of this glycolysis pathway or EMP pathway. So we started with glucose, we went 10 steps, and our end product of this glycolysis is pyruvic acid, which is a 3-carbon compound. So we got two molecules of pyruvic acid. So first, the energy here in the first step we are using 1 ATP. In the third step we are using an ATP. Here in the sixth step we got 2 NADH2. First time we are getting energy. In the seventh step we got 2 ATP, and in the 10th step we got 2 ATP. Okay, I hope you have understood till now. So this is the seventh step, this is the eighth step, this is the ninth step, and this is the 10th step. So glycolysis is a 10-step process. From glucose, we are coming to the two molecules of pyruvic acid. So in the first and the third, we are using an ATP, whereas from the sixth step, we are getting energy. So in the sixth step, we got energy in the form of NADH2. In the seventh step, we got 2 ATP, and in 10th step, we got 2 ATP. Okay, so here it is written that step 1, 3 and 10. This 1, 3 and 10 is an irreversible reaction. Means it cannot reverse. It cannot go back. It is a forward reaction only. And now comes this fructokinase enzyme. This enzyme is known as pacemaker. Why we are calling it? Because it maintains rate of reaction by analyzing the amount of ATP or ADP plus inorganic phosphate present in cytoplasm. Based upon the concentration of ATP or ADP plus inorganic phosphate, this process can move forward with the help of this enzyme. If this enzyme is recognizing that ATP is less in the cytoplasm, then the rate of forward reaction will increase. Whereas if it is recognizing that in cytoplasm ATP is more, then the rate of reaction decreases. Okay, so that's why we call phosphofructokinase as pacemaker. And as I told earlier, in the sixth step, we are getting energy in the form of 2 NADH2. Here, first time we are getting the energy. In the seventh step, we are getting this 2 ATP. And in the 10th step, we are getting 2 ATP. So I hope you have understand this process, glycolysis, how we are moving forward from glucose to two molecules of pyruvic acid. Okay. The term glycolysis has originated from Greek word glykos, which means sugar, and lysis, which means splitting. Here, glucose is being splitted into two molecules of pyruvic acid. Okay. The scheme of glycolysis was given by Gustav Emden and Otto Meyerhoff and J. Parnath. So that's why we call it as EMP pathway also. In all living organism, whether it is aerobic or anaerobic, the first step in the cellular respiration is partial breakdown of or oxidation of glucose into two molecules of pyruvic acid. Whether it is aerobic respiration or anaerobic respiration, glycolysis will be there in each and every respiration. Okay. The glycolysis is a common phase between aerobic and anaerobic respiration. In anaerobic organisms, it is the only process in respiration. So, in anaerobic organism, only glycolysis will happen, whereas in aerobic organisms, this glycolysis will be followed by several other steps also, because we have to come. We are in anaerobic, we are completely broken down that glucose into carbon dioxide and H2O. The glycolysis occurs in cytoplasm of the cell. This you have to remember, and this process is independent of oxygen. It means it can occur in both conditions. If the oxygen is present or absent, it doesn't matter. Glycolysis will take place because it is also taking place in aerobic and anaerobic condition also. Okay. So it involves a series of 10 biochemical enzymatic reactions in cytoplasm. We studied all the 10 steps in the previous slide. Okay, I hope you have got till the point. Now let's move to the next slide. Now comes the link reaction. Okay. So what is link reaction? Link reaction is also known as gateway reaction, transition reaction, or oxidative decarboxylation reaction. This all the three names you have to remember. And site for this link reaction is mitochondrial matrix. Okay. So here what happens, two molecules of pyruvic acid which are formed in glycolysis will undergo this changes and will form two molecules of acetyl coenzyme A. In presence of pyruvate dehydrogenase complex, here one NAD plus molecule will be utilized and two NAD, actually it is 2 NADH. 2 NADH plus will be used and 2 NADH2 will be formed. Here also we are getting two molecules of NADH2 as energy. And here the first time CO2 is getting released. In glycolysis, we haven't seen carbon dioxide removal. Here we are seeing the carbon dioxide removal. So first time the carbon dioxide is getting released here in link reaction. And this two molecules of acetyl coenzyme A. This acetyl coenzyme A is the connecting link between glycolysis and Krebs cycle. How it is connecting link, we'll see in the next slide. So this complex which is being used is consist of this pyruvate dehydrogenase complex. It is having magnesium ion, thiamine pyrophosphate, coenzyme A, lipoic acid, and NAD+. This you have to remember. What are the components of pyruvate dehydrogenase complex? Okay. So here in link reaction, nothing is happening. Only two molecules of pyruvic acid is getting converted into two molecules of acetyl CoA, and this acetyl CoA will be used in the next step, which is Krebs cycle. So that's why we are calling it as connecting link between glycolysis and Krebs cycle. So let's move to the next slide. Now this process connects glycolysis and Krebs cycle, so it is called link reaction or gateway reaction. The name we are calling it as link reaction because it is linking glycolysis and Krebs cycle together. During this process, the first time CO2 is evolved during respiration, because in glycolysis we didn't study that there is any CO2 removal. So first time we are seeing that CO2 is removed in link reaction. This acetyl CoA is a connecting link between glycolysis and Krebs cycle, as I told earlier. Decarboxylation and dehydrogenation, that is oxidative decarboxylation, takes place during the formation of acetyl CoA. Okay, here the decarboxylation as well as dehydrogenation is taking place. Acetyl CoA is formed in the matrix by enzyme pyruvate dehydrogenase complex we studied in the previous slide. This reaction requires 5 cofactors, that is magnesium, lipoic acid, thiamine pyrophosphate, NAD+, and coenzyme A. Okay, let's move to the next slide. Now comes the Krebs cycle. This cycle was given by Hans Krebs, as that's why we are calling it a Krebs cycle. It is also known as tricarboxylic acid cycle or TCA cycle or anapleroptic pathway. This you have to remember. It is also known as anapleroptic pathway or tricarboxylic acid cycle. Okay. It is also known as citric acid cycle or C6 cycle because the first compound which is forming in this uh, cycle is citrate, which is a 6 carbon compound. So that's why we are calling it as C6 cycle. It is also known, it is also a 10-step cycle. The site for this cycle is mitochondrial matrix, as in link reaction also. Link reaction was also taking place in mitochondrial matrix. And here, all enzymes of TCA cycle found in matrix of mitochondria except this succinic dehydrogenase. This you have to remember. Thus, all the enzymes which is being used in TCA cycle is found in matrix of mitochondria or mitochondrial matrix except this succinic dehydrogenase which is found in cristae. Cristae is that fold which I talked about. This kind of hole was there na, in inner mitochondrial membrane. So this is cristae. In cristae, this succinic dehydrogenase enzyme is found. Now let's study the Krebs cycle in detail. Okay, so this link reaction we studied. Wait a minute, I have to change the color. So this link reaction we studied earlier, pyruvic acid, a 3 carbon compound, is getting converted into acetyl-CoA, and this acetyl-CoA is used is reacting with its oxaloacetate, that is a 4 carbon compound. It is combining with 2-acetyl-CoA and it is forming citrate, that is a 6 carbon compound, and this citrate is in presence of aconitase is getting converted into this cis aconitase. Okay, this cis aconitate is getting converted into isocitrate, and this is getting converted into oxalosuccinate, and this is getting converted into alpha ketoglutarate, and this is getting converted into succinyl CoA, and then succinate, then fumarate, and malate. These are the uh, compounds which are being formed in this cycle. So first is citrate. Citrate is forming, which is a 6 carbon compound. That's why um, we are calling it as C6 cycle. And this citrate is getting converted into cis aconitate in presence of aconitase enzyme. Then this cis aconitase is getting converted into isocitrate, then oxalosuccinate. This isocitrate is getting converted into oxalosuccinate. Here, one energy molecule is being formed. Here, NADH2 is formed. So, here, first energy we are getting in this cycle. Now, this oxalosuccinate is getting converted into alpha ketoglutarate. Okay, here one removal of carbon dioxide is taking place, and this oxalosuccinate was a 6 carbon compound, whereas alpha ketoglutarate is a 5 carbon compound. Here one coenzyme A will be added and one CO2 will be out, and here also one energy is formed, that is in the form of NADH2. Okay, now because one more CO2 is released here, so succinyl coenzyme A, which is a 4 carbon compound, is formed. Now here one GDP plus inorganic phosphate is used, and one GTP, which is equivalent to one ATP, is formed. And here whatever the coenzyme A which was used in this step, the conversion of alpha ketoglutarate and succinyl CoA, is getting released in this step, where after releasing coenzyme A, this succinate is formed. Again, from like this is also a 4 carbon compound, this succinate, and this succinate will get converted into fumarate, which is a 4 carbon compound. In between this, one energy will be formed in as FADH2. Okay, and then this fumarate is getting converted into malate, and this malate is again getting converted into oxaloacetate, and during this conversion one more NADH2 is formed. So in this process, which step we are getting NADH2, in which step we are getting energy is So first we are getting energy here. Mhm. First time we are getting energy here, okay, because here one NADH2 is being formed. Again we are getting here one more NADH2 is formed. Here we are getting one GTP which is equivalent to ATP, okay. Then again we are getting FADH2 in conversion of succinate to fumarate, and then NADH2 here when malate is getting converted into oxaloacetate, okay. So how many NADH2 we formed? 1, 2, 3. 3 NADH2 we are getting. See here. 3 NADH2 we are getting. And wait a minute. One FADH2 here. While conversion of this succinate to fumarate, we are getting one FADH2 and one ATP, which is in the form of GTP, which is equivalent to ATP. So we are getting one ATP. So during one Krebs cycle, we are getting 12 ATP. Okay. So if one glucose is getting converted into 2 pyruvic acids, so 2 acetyl coenzyme A will be there. So 2 acetyl coenzyme A is there means 2 Krebs cycles will be there. So if 2 Krebs cycles are there, then if we are getting in one Krebs cycle we are getting 12 ATP. So in 2 Krebs cycle we'll get 24 ATP. And here this one FADH2 is equals to 2 ATP. Whereas one NADH2 as I told earlier is equals to 3 ATP. Now this compounds, how you will remember, like which compound is getting formed after which compound. So one trick is there. Okay. So first compound is this citrate. So we can call it as C. Okay. Now the second we are getting is isocitrate. So we can call it as I. CI, no. No is this alpha keto. So C I know some, that is succinil. So C I know some simple formula, that is fumarate, making, that is malate, options, that is oxaloacetate. So this is a trick. See, I know some simple formula making options, that is citrate, isocitrate, alpha ketoglutarate, succinyl coenzyme A. Simple means succinate, formula means fumarate, making means malate, and options is oxaloacetate. So that's how you can learn the this cycle. Okay, this is a short trick. So see, I know some simple formula making option. So one Krebs cycle is result in the in the synthesis of 12 ATP. So because one glucose is forming two acetyl coenzyme, so two Krebs cycle should be there. So 24 ATP is formed. Okay, let's move to the. Now comes the questions. So in this lecture, we have studied what is respiration, the types of respiration, that is aerobic and anaerobic respiration. Then we studied glycolysis, where it is taking place, what are the steps, so 10 steps are there. So what are the enzymes which are being used there, how the steps are there, I have explained it to you. Now then we studied link reaction, which is a connecting link between glycolysis and Krebs cycle. And then the third cycle we studied Krebs cycle. Okay. So I hope you have understood till the time. Now let's start with the questions. So first question is which step is called gateway step or link reaction in the aerobic respiration? So in aerobic respiration, which step is known as link reaction or gateway step? Glycolysis, no, this cannot be. Formation of acetyl coenzyme A, this is correct answer. Why? Because in this pyruvate, two molecule of pyruvate is getting converted into two molecule of acetyl CoA. So the formation of acetyl CoA, and this reaction we are calling it as link reaction or gateway step. So these kind of question will be asked. Okay, easy questions but conceptual. Now comes the match column 1 and column 2 and select the correct option from the codes given in below. So here is the column 1 and here is the column 2. So glycolysis, TCA cycle and ETS. So here we have to match where it is occurring. Okay, so glycolysis, as we studied, takes place in the site of glycolysis is cytoplasm. TCA, it occurs in mitochondrial matrix, whereas ETS, that is electron transport system or chain, this occurs in inner mitochondrial membrane. So if we have to pair, is A3. So here A3 is given in A and B option. Now comes B2. So the second option will be correct and C1. So this is how the question will be asked in JRF examination. So you have to study just based on the type of question which are being asked. Okay. Next is how many molecules of ATP are produced per molecule of FADH2. As I told, one FADH2 will result in the formation of 2 ATP. How they will form, we will study in the next lecture. And one NADH2 will result in the formation of 3 ATP. So here we have been asked only FADH2. So 2 ATPs are formed. So option B is correct. Okay, I hope you have understood the lecture properly. And if you are coming for the first time, so I would request you to first watch the first lecture, that is transport in plants. So from that, if you will start, you will get the concept what I have taught in this lecture. So thank you for watching and keep watching Agri Addict. Thank you so much.